Hello, everyone. I'm Christy Rizk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is PGX Beyond Drug-Metabolizing Enzymes, a Multigene Approach to Predicting Drug Response, and our sponsor is agena-bioscience. Our panelist today is Dr. Ryan Sprissler, Lead Scientist at the University of Arizona Genetics Core for Clinical Services and Co-Founder and Chief Scientific Officer of Geneticure. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar screen. If you look to the tray at the bottom of your window, you'll see a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Dr. Sprissler. Please go ahead.
Thank you, Christy. I appreciate the introduction. So I want to get a few quick thank yous out of the way. I want to thank GenomeWeb as the host today. I watch a lot of these webinars and have really enjoyed them. And I thank you for letting me have the opportunity to speak today. I want to thank Agena as the sponsor and also for being such great partners in the development of our technology. And also thank all of you for listening today. I know it's a lot to take an hour out of your day. Thank you for that. And then I also want to make one quick disclaimer. As indicated in my bio, I do have an appointment here at the University of Arizona, where I lead the genetics core facility, and I'm a founding member of the Center for Applied Genetics and Genomic Medicine. For the purposes of today's talk, I'll be speaking in my capacity as Chief Scientific Officer and co-founder of Geneticure. So I'm going to be wearing my Geneticure hat today. So a brief overview of the talk today. As the title indicates, I'm going to be talking about a multi-gene approach to predicting therapy response. I'm going to be doing this in the context of hypertension, one of our assays. And so I'm going to talk a little bit about hypertension. I'm going to give a kind of a description of our panel and the algorithm we're using. I'm going to talk briefly about our testing platform and validation. And I'm going to then go through some of our clinical studies and clinical trial data, and then wrap up with a brief description of some of the health economic studies we've done. And then we'll open it up to questions at the end. So at Geneticure, we kind of came up with a mission to use the genetics of integrative physiology. I'll kind of explain a little bit more what that means, to eliminate trial and error in medicine. And, you know, the trial and error term, I guess, is probably a bit harsh. I would say, you know, today a lot of these standard of care treatment algorithms are actually, you know, they're using population study data, they're using epidemiological data to inform some of their treatment algorithms, but that is really a far cry from treating the patient in front of you and us as individuals. And as we'll kind of see, it really leaves a lot to be improved. And so we've taken a bit of a different approach. Again, you know, as the title of the talk kind of explains, you know, I hope it isn't disparaging to the drug-metabolizing enzyme, the DME panels that are being used in pharmacogenetic testing. Those are important. It is important to know how you're metabolizing these medications, how they're getting into your bloodstream. But at a certain point, we also need to know what are the targets of those drugs and are they effective? And so in complex diseases, especially one like hypertension, we have a number of different disease states that are in various stages of development, our development pipeline. But I'm going to be talking about our hypertension assay. It is, you know, it's a disease state that has multiple classes of drugs that have multiple targets. And so we'll kind of see why that's important and why that it makes sense to use not just DMEs, but also some information about the functionality of each organ system. Another thing, another kind of differentiator for us is this evidence-first approach. Most of us at Geneticure come out of academia. And so we've spent the last number of years just generating clinical evidence, generating data, publishing in peer-reviewed journals. It's important for us to get this out there and make sure that it's on solid footing as we stand up testing like this. And then also we've done a lot of patent work. We were recently awarded a patent for a hypertension assay, largely around protecting some of our algorithms and a bit of our process. So As I said, we're going to look at hypertension. I'm not a clinician. When we started this work 6-7 years ago, I was shocked, frankly, at how this disease is treated, especially for a disease state that roughly 1 in 3 Americans will experience in their lifetime. And as we'll see, the standard of care, I was a bit shocked at both the treatment rates and control rates, which we're going to look at. But it's also a very expensive disease, and it's also treated with trial and error. So we feel like this is a good, a big opportunity to kind of improve care and really drop costs. So hypertension as a risk factor is obviously, if we're looking here, deaths attributed to risk factors, it's the leading risk factor. It's across the board. This isn't a rich person disease. It isn't a poor person's disease. It truly affects everyone. And while it affects all people across the spectrum, it affects certain populations disproportionately. And so this is where, looking at treatment rates, we're at 74, 70, and 60% treatment rates for whites, African Americans, and Hispanics, respectively. It's not crazy terrible, but it's not very, very good either. And the control rates are where actually the story gets much worse. So, roughly 43, 37, and 31%, again, for these populations, respectively. That is actually quite terrible. I should mention for this particular study, this is from using the JNC7 guidelines. This is the standard of care. The JNC is a Joint National Committee 7, where they're now using, there has been an upgrade to this, the JNC 8 guidelines, which have had an appreciable increase in control rates. But still, even if we're being generous, we're up to maybe 60, 50, and 40%, something like that for these populations. That's still pretty terrible and leaves a lot of room for improvement. So what am I talking about with this integrative physiology approach? Well, high blood pressure is much more of a symptom, so to speak, that can have a number of causes. One or any of these organ systems can cause, one or more of these organ systems can cause a problem. We have cardiac output, which is how hard the heart beats and the general heart rate. Your genes are going to impact this, the functionality of your cardiac system, and beta blockers are the class of drugs that are going to be used to treat this organ system. Your kidneys, sodium reabsorption, your genes are going to impact sodium handling in your kidneys, and diuretics are going to be the treatment used to target that organ system. And then there's your vessels, your vasoconstriction, your vasculoconstriction, and sodium reabsorption there. Vasodilators are going to be used to treat that organ system, and again, your genes are going to impact the functionality of that organ system as well. So we have multiple organ systems involved and multiple classes of drugs that can be used. And, what is the problem with the standard of care, aside from making this gentleman pretty unhappy? we have a standard of care treatment algorithm, which is basically going to start with, in most cases, a diuretic. We do understand that vasodilators, in some cases, are moved to a frontline therapy. But in general, we're going to start with monotherapy of a drug. In this case, we'll say a diuretic. We're going to monitor for 3 months to see if this drug is working. Is it effective? If it is not getting your blood pressure under control, this is an important distinction. They don't remove you from diuretic therapy. They'll actually layer on the next set of class of drugs, a vasodilator in this case, and then we're going to wait another 3 months. So now you're on 2 drugs. It's been 6 months, and if your blood pressure still isn't under control, they're going to move to a third class, the beta blockers. And so at this point, you're on a number of medications. Your blood pressure may or may not be under control. They can always go back and reinitiate with a dosage adjustment, but in most cases, you're going to frustrate a lot of patients here. And so this, and again, organ damage is occurring from the moment that the patient is experiencing hypertension. So during this entire time, organ damage has been occurring as you haven't gotten these under control. And this layering effect of drugs is going to have a major impact on adherence rates. So as you layer these drugs, your non-adherence is going to skyrocket. And I think we kind of all know this intuitively and kind of, heard it anecdotally where people get frustrated. My doc doesn't know what he's doing. I'm on a bunch of drugs and they're not working. And so they either just stop taking the medications altogether and drop out. They stop going to office visits. Whatever. You've created a serious problem here. Aside from that, a lot of times, we consider these drugs to be fairly safe, and they are. They're all approved drugs. But there is a bell curve response to these drugs that I think goes a little bit underappreciated. This is response to hydrochlorothiazide monotherapy. This is a diuretic. And for it's not bad enough that it may not be effective. But in some cases, in this case, in this study, a well-controlled study, 20% of patients saw an increase in blood pressure. So if the medication isn't working, it's not just, well, that's no big deal. There are consequences to this. In many cases, in some cases, you're making this worse. And again, to kind of highlight the problem with layering and being on a number of ineffective drugs, there is a side effect profile for each of these drugs. And again, they are approved drugs. They're good drugs, but there are consequences to administering these. And the content of this slide isn't necessarily important, but just to understand that there are significant side effect profiles for each of these. And, we've heard in some cases the argument, well, just, put everybody on all the drugs. What's the problem here? Just, over-medicate, right? They're not super expensive drugs. Well, let's just throw let's throw the book at it. Well, again, non-adherence is a huge issue, and side effect profile becomes, people are just going to stop taking drugs. If you're on an ACE inhibitor and you have the ACE cough, you're going to stop taking those drugs, even if they're effective. Even worse, if they're not, you might drop out of treatment altogether. So, again, it's a problem and leaves a lot of room, again, an opportunity for improvement. So the standard of care, aside from being inefficient, can also be dangerous, and that definitely needs to be recognized. And there's a number of reasons. There's a number of problems kind of inherent in this standard of care. Average response rate to each medication is only 50%. So we have these different classes of drugs. Response rates aren't across the board. We have a number of visits. If we can decrease the number of visits, that's good for docs. That's good for patients. And this layered medication approach, as I kind of discussed, is working against us. And also, I'm not going to get too much into this, but there are a lot of studies. There's more and more reports coming out of really long-term side effects of use of these medications, and you don't want to risk any long-term side effects for medications that may or may not be ineffective, and that's where we're at now. Now, what is kind of our solution to this, or what have we developed to kind of address these issues? The Geneticure hypertension assay. This is kind of a quick overview of the process. Upper right here is the test kit itself. We use a buccal swab collection. So cheek swabs. After you swab the cheek, there's a little plunger. You eject the comb into these vials full of lysis buffer. So we've lysed the cells at this point. They are non-biologic. They can go through the mail. They're put into this kind of padded mailer, mailed off to the lab. So it's a pretty simple process, and patients or docs can do this rather quickly. And just real quickly here, again, physician orders the test. We do buccal swab collection, do a genomic extraction, run some PCR reactions. I'll talk a bit about that, get some genotype calls, run them through an algorithm, and create a report. Over here on the right is our report. We found it really important to make this a very clean, simple report that docs can get information from quickly. Again, not to be disparaging about kind of the pgx testing, the DME testing. I'd say with the exception of some of the psychiatric pgx tests, these can be, upwards of 20 pages deep, where, you're getting a lot of extraneous information and aren't always there at the point of care. So, what we're trying to do is put the test just after diagnosis, just prior to treatment. So we want this to be kind of an actionable report, something they can use at point of care, versus kind of just your drug metabolizing profile that for a bunch of drugs that you may take down the line. I do want to really reiterate our intended use here. This is meant to be a decision support tool. So this is for it's another tool that the clinician can use to initiate treatment or have more information on treatment. The patient is going to know, or the physician's going to know more about the patient than we are. They're going to know comorbidities. They're going to know if they're diabetic status, if they're an epileptic, if they're on other drugs that might interact with our primary recommendation, or there's a number of reasons why the therapy the frontline therapy that we would recommend might not be appropriate for that patient. And again, the physician's going to know that. So this is decision support. This is another tool for the physician to use. And I would also say that there's no reason for a clinician not to have this information, and really offers them a chance to kind of interact with the technology and again use our report to help prescribe. So how did we build the assay? This is kind of a funnel here. We started at the top. We wanted to select SNPs. We had a number of filters. So this was the initial SNP selection was a huge part of the process. It took a very long time. This is a lot of primary literature searching, a number of studies, if they were clinical studies or preclinical studies, animal models. We just wanted to find genes, and particularly variants, that had a high association with organ functionality or specific therapy response. So, the cliche of siloed research was really evident here. A lot of researchers were studying one gene, one variant, and how it affected these organ systems, how it affected hypertension, how it affected therapy response. What we wanted to do is go across and aggregate all of that information, but also have some cutoffs. And so we wanted to make sure that each of the variants we were selecting had a reasonable MAF, a minor allele frequency over 10%. So we wanted to select things that were common in the general population, and we wanted them to be highly functional. So they had to be common and functional. We fully recognize we're not testing rare alleles. These are hard to clinically annotate. There's not a lot of evidence behind them. There are a lot of cases private variants. But we understand that there could be a number of those in the population. They're going to have a large impact, but we're not looking for those. We're kind of casting the widest net possible. So from there, I'm going to talk a little bit about, and a little later on, talk about our trial data, but we ran a small pilot trial, and this was to test the individual genes. What do we want to keep? What do we want to throw out? What is, in fact, functional? And from there, we refined the panel. We ended up removing 2 SNPs that we originally had in there because we weren't seeing any evidence that they were actually functional or had any contribution to therapy response. We ended up adding 5. I'll say between steps 1 and 3 here, there was a number of years, so more information had come out, so we added that in, and we landed on 17 SNPs in this particular panel. So from there, not all these variants are created equal. we had to weight the SNPs. So some variants have a larger impact on functionality. Some have more strength of evidence. We want to make sure we're taking those things into consideration and really balancing this out. We want to then use the weighting of these in an algorithm that's going to help weight organ systems against each other, so we can look at organ system functionality and have some information on where to start treatment. If some functional threshold is not met for that particular organ system, we're going to move to the individual SNPs and haplotypes. But in general, this is kind of the end point. We then wanted to test this algorithm on a larger population. This was our large trial, our large retrospective trial, which I'll talk a bit about. And then we wanted to take that data and use machine learning and some AI techniques to kind of tune the algorithm and also make sure that kind of all the inputs and outputs are matching up. And I'm going to talk a little bit about that as well. So first part of this process was, as I mentioned, was to go through and look at the contribution of each individual variant and make sure that we've selected the right variant. So this comes from previous studies, and this kind of went into our weighting process. So we're looking at the difference between individuals who carry the functional genotype and the non-functional genotypes and are on the target drug. So if we stay over here on the left in the cardiac system and we look at ADRB2, so this is the adrenergic beta receptor 2, amino acid position 16. If we have two patients, one with a functional genotype at this position, they're going to see a roughly 4% drop in millimeters of mercury versus a patient who has the non-functional genotype who is also on a beta blocker. So as you can kind of see, again, not all of these variants are created equal. Some have a higher impact on functionality. If we look at CYP2D6 here, this is one of the DMEs, kind of underlining my point I made earlier. While they're important, they don't always have an effect on the outcome variable. So in this case, the non-functional versus functional genotype on the target drug and beta blockers are metabolized to the CYP2D6 pathway. We don't see any difference in baseline millimeters of mercury. So again, it's important, but not the whole story. From here, we wanted to do some sorting and weighting of the variants. What we're looking at here is actually the bottom half of our clinical report. We wanted to bin the variants into organ systems. So we've binned them into the cardiac, renal, and vasculature systems here. And then we wanted to use genotype to assess functionality. In some cases, as we see here with ADRB1, position 389, third one down, the heterozygous status is enough to give a fully functioning genotype. In some cases, if we look at ADRB2, position 27, the heterozygous genotype actually infers an intermediate function status. And in other cases, the heterozygous status is non-functional. So we kind of have this stoplight, red, yellow, green system to assign functionality to each variant. Then we combine that, bin that into an organ system. And then on the right here, we basically use the weighting to produce a likelihood of response in the bar graph between different drug classes. And so this is giving you kind of an individual contribution of functionality, but then a larger kind of aggregated functional assessment of that organ system. What we wanted to do after that process is to use some AI techniques. It is a complex genetic profile. 17 genotypes may not really seem like that complex, but we do have an interconnected physiology going on here. There's a lot of complexity introduced in the multiple drugs that the patients are on and the variable responses. So in our retrospective studies, what we're doing is using chart review data So we have patients who enroll in these trials. They've been, they've gotten their blood pressure under control, and we have a history of how they got there. We genotype them, and then we kind of use those as input variables. And we also have gene-to-gene interactions. So, we were doing this by hand, and we wanted to make sure that we've weighted things properly and that we've kind of, accounted for at least the majority of the complexity. But we wanted to try to solve for that complexity also using some of these AI techniques. We used random forest classifier techniques to build multiple algorithms. We then, as I said, use the clinical trial data as training sets. So we put all that demographic data, all the drugs administered, the order, the timing, the change in blood pressure over time as these drugs are administered. We also have age, weight, smoking status, all this kind of stuff. So we have a lot of data that we can use as a training set. And then, we've also added genotypes in. And then so we kind of set all these AIs loose, all these algorithms loose on this training data. And then we built an environment we codenamed Thunderdome, and had them all kind of battle it out to see which one and which of these AIs ended up being kind of most predictive. And so you're seeing a bit of the results here on the left. Are the results of these AI-generated confidence scores. So we ended up with about 110 trees assigned to a set of random set of 6 genes, and in the end, we get these confidence scores. So in the box, if you're looking on the far right, your effective WP. This is kind of a confidence score for each class of drugs. And this was kind of the John Henry test, right? We had the human-derived algorithm where we best we could do with the information given, and then we kind of had this true machine kind of going after the same endpoint. And what was really interesting and really great to see is actually this was really self-confirming. So in the majority of cases, they were matching. So we kind of knew things were headed in the right direction. So what does that end up producing? Well, we get a report here. I showed this a bit earlier, and we looked at actually the bottom half of it. So on the right is the full report. The top half is kind of the output. Again, we want to make this something that it can be given at point of care and can be used easily and quickly. We wanted to give primary recommendation, secondary recommendation, and just go down the line. Again, I want to reiterate, we are not recommending the use of any drugs that aren't already approved for hypertension. These are drugs that would be administered anyway. We're simply shuffling the order using this genetic functionality to inform how we shuffle that order. And then below each recommendation, we give a general narrative that the physicians can kind of go through if they want to learn more. We also recommend if they have any questions that they contact us, and we can walk them through things as well. So here I want to switch gears a little bit and go through the platform and kind of how we developed the test before I get into the clinical studies. The MassARRAY was really a great fit for us. Our search space of 17 genes, we wanted to make this, we wanted to lean it out so that it was as cheap as possible, so we can get this out to the most people possible, and really, it's a signal to noise kind of issue. So we wanted to keep it really focused on these really functional and predictive variants. So the number, we're not doing a million SNP chip kind of thing. So the number of variants we were testing fit really nicely with the MassARRAY platform. Also, the cost was really great as well. It allows us to batch and drive costs down. We had they're not all SNPs, so we are looking at one indel in the ACE gene, the angiotensin converting enzyme. It's a roughly 280 base pair insertion. And so it was great to have the flexibility of, we can use TACMAN assays to look at all these SNPs or something like that, but indels pose a problem. We want to get it all in one assay. and that can be very difficult using other technologies. So to have kind of be able to detect all these different variants was quite nice. For those of you who don't know how it works, the MassARRAY is effectively a mass-derived genotype instrument. It's a MALID-TOF at kind of the core of it. So what we're doing is we're doing a multiplex PCR up front. So we're using one reaction to generate all of our products. And then there's a second step, which is the extension reaction, and effectively we're putting a primer down just proximal to the SNP we're testing. We do a single base extension that extends the base into the SNP because nucleotides all have different masses. Once we put this through the MALID-TOF, we end up getting a peak, and we're able to, again, use mass to derive the genotype. So it's a pretty slick little instrument, and has worked out quite well for us. And then, the turnaround time is important. We can't have a ton of hands-on time, so it's also been a nice technology for us to use to kind of minimize the turnaround time. And I will say it shouldn't go understated that the data analysis piece of this is quite nice. We can set thresholds such that if the genotype call meets that threshold, we have a very high confidence. If it doesn't, it's queued for rerun, and we don't have to spend a ton of time picking apart the raw data. So again, the data analysis piece of this technology is quite nice as well. The analytical validation we ran wasn't too difficult. We across the top here are all of the variants we're testing for, and down the left-hand side are a number 22 chorial cell lines that we use. We wanted to make sure that we were representing each of the minor alleles at least twice. And that's important for make sure that we're able to do full detection of all of these variants. And then we took all of these cell lines and ran them through other technologies for verification. We used TACMan, we used Sanger sequencing. So we really had a rock solid truth set here, samples to run a lot of our validation studies, specifically our analytical validation studies. And then we also use these for batch testing of actual samples. We use these as controls that kind of ride along with the processing of batch samples. So the reproducibility was really great. Another kind of testament to the agena MassARRAY. We did technician to technician, day to day, chip to chip. There's two formats, this 96 and 34 well format. The volumes are slightly different, so we want to make sure we were getting consistent results across those formats. And then we have multiple instruments in the lab, and we went instrument to instrument, and again, performed really well. The assay performed as good as we could have hoped. Here's a bit of those results. We tested a total of 2,816 genotypes, and we're really quite happy with the results. Our sensitivity, specificity, positive predictive value, accuracy, all those things were fantastic. The NC, our no-call rate there, we're at about 96%. I want to mention not all no-calls are created equal either. If we look across the top here and we look at our no-call row, you'll see a lot of sevens in there. And basically what that's indicating is that if the sample produces, we're not producing a bunch of incomplete profiles. So we don't have a bunch of holes to plug. And that's really important. That's going to be much more difficult than if we run a sample, it either works or it doesn't. So there were 7 samples, as you can kind of see here, that didn't produce a profile at all. So, and then there's a few that, are a little more prone to be of lower quality. Usually this has to do with fragment size and a bit of the design. But in general, the sample either works or it doesn't. It's not giving out these incomplete profiles, which is very nice for us because it's a little more difficult to kind of plug all these holes. So I'm going to move now into our current clinical data and research studies. And we have, I'm going to give my email address at the end of the talk and a link to our website. I'm only going to touch on some of the highlights and bullet points of these studies and these trials. But again, we've published a lot of these, and if there's any specific questions, I would refer you to those publications. I'll show them in a minute. But for our clinical trials, we started, as I said, with that rather small retrospective study. So we selected all of our variants. We wanted to do a proof of concept trial. This was a chart review where we looked at the individual contribution of each variant. That was completed some time ago. And then through our clinical partners, Fairview Health up in the Midwest, we did that pilot RCT. This was basically an inferiority study I'm going to talk a little bit about, but this was prospective, a randomized controlled blinded study where we had 2 arms, a standard of care arm and a Geneticure arm. And then we did a larger retrospective trial, and this was a bit of a typo here on the number of subjects. It's actually a little under 400, not 700. But again, this was to test the algorithm and really kind of fine tune our results. And then lastly, I'm not going to go into this, but we have a large multi-center trial right now that is, we've kind of just finishing up the trial protocol design. This is going to be a prospective, we did some power calculations, basically determined we're going to need about 1,000 patients, 500 or more in each arm. We'll be recruiting that study soon. And again, a multi-center trial. This will be kind of the gold standard for our clinical evidence. And each of these trials we're kind of building on the previous data so that we can get to this kind of large, really expensive trial where we look at a number of different endpoints, and the ones we're particularly interested in are reduced time to control, lower absolute blood pressure, are we using fewer meds, what does adherence look like? So we'll get a lot of really rock-solid clinical evidence from this trial particularly. So again, very important to us that all of this stuff is put out into the peer-reviewed community. These are our publications. Again, I'm only going to be touching on some of the results, the kind of highlights. If anybody has any further questions, please go out to these publications and read about these studies. We also did a health economics publication that I'm going to get into a little bit as well. But these are available to anybody, and I can also, I'll give my contact information. Feel free to contact me, and I can get these in your hands. So our first retrospective study, this was that small proof of concept, testing the genotypes. Did we pick the right ones? And what we're looking at here is the change in diastolic blood pressure in patients with the functional genotypes who are on beta blockers versus patients who are not. So this is kind of asking the same question in a little bit different way. So we have, if we're looking at, let's ignore CYP2D6 here, but if we're on ADRB1 position 49, and we have 2 patients who are both on beta blockers, one has the functional genotype and one has the non-functional genotype, what's their difference in blood pressure? And this is really giving us a good idea from this chart review data if we've selected the right variants. And in this case, it looks like we have. So we're fairly confident, actually, that we have, I'm sure other things will come to light in the future, but I think we have kind of the main players selected, the ones, the highest impact, the most informative. I think that we've, exhaustively searched and found those. But in this case, if we're just looking at, this is just showing the beta blocker response, but we're looking at an average decrease of about 5 millimeters of mercury for patients on a beta blocker versus not when they have the functional genotype. And so this may not seem like a huge drop in pressure, but actually this is wildly important. We did not measure this directly, but 4 to 6 millimeters of mercury drop in epidemiological studies is associated with a 40% reduction in stroke and 26% reduction in cardiovascular death, heart attack. So this is a big deal. We're not kind of nibbling at the edges. These are big impacts. Our pilot RCT trial, this is an inferiority study. Here, what we wanted to look at is, okay, are we, we want to make sure that we're at least meeting some of the metrics of the standard of care, even though it's not great. Are we falling short in some areas? And in this case, yes, we might get some folks to control quicker, but, after 6 months, are we meeting where the standard of care is at? And we were right within range there. So very happy to see that, maybe the standard of care gets to the right drug eventually, but, did they do a better job in the end? And the answer is no. So that was good to see. We're using slightly fewer meds. And the big one we want to highlight here is the standard of care arm had many more serious adverse events. And in this case, and again, these are admittedly a very small RCT trial, and we're going to get more information in our larger upcoming trial, but 70% of the severe adverse events occurred in the standard of care arm. Most were due to diuretic side effects, and some of them required hospitalization. So this is wildly important for kind of the health economics piece here, because a lot of the savings is going to be found in limiting or lowering these serious adverse events, which are incredibly expensive to treat. So the third trial, which was our second retrospective trial, this was, again, testing the algorithm. We want to look at difference in blood pressure from diagnosis to current one-year average if patients are on the first drug that we recommended. Again, each trial is kind of building on the evidence of the previous, so we wanted to say, okay, we've selected the right variants. We're meeting the standard of care, bar, and then, how is our algorithm performing? And again, our main concern here is on the far right, our delta map. This is mean arterial pressure. And we're looking at, again, average mean arterial pressure, 5.6 millimeters of mercury. This is 36% lower blood pressure if the patient had been prescribed our first recommended drug. So again, a lot of evidence trending in the right direction. We're building on this, and we really feel like carrying a lot of momentum here. So just to kind of summarize the clinical evidence we've done, we have 3 trials, a 4th on the way, a total of a little under 700 subjects. We have a number of publications, and we're starting to now present some of this data at conferences. But big takeaways here, patients under control within 6 months. We're really getting folks under control. 30% reduction in blood pressure versus standard of care. That's what I just showed you. Less adverse events. Again, this is wildly important to both just treatment, but a lot of the health economics, and that's a good segue into the kind of cost of care here. 40% reduction in cost. And so the health economics piece of this is rather dramatic because of the size of the hypertensive population in this country. So you have, even small changes in cost of treatment on a per patient basis are going to extrapolate out to massive numbers because of this massive population suffering for hypertension. So this was our publication. We have a simulation study we ran. I want to mention importantly here that the simulation study we did was informed by the clinical trial data. So we didn't just kind of make things up. We used our retrospective trial data to actually inform the model. So this is as close as we could get to real-world implementation of our tests. And if we're looking here, 3-year cost savings of 40%, which turns to $42 billion in an insured panel of 10 million patients. So that's, again, these numbers kind of get crazy due to the population size. We made, in our health economics simulation, we made some general assumptions on the population. On the left here, you're looking at the standard of care. This is not an assumption. This is actually kind of the real world. Usually it's about 5 visits. It's going to take at least 2 meds. And we have 60% under control. 40% treated, uncontrolled. Can't do much about the untreated. Hopefully, I would say, hopefully with more kind of awareness, more tests like this out there, some of these untreated folks might want to enter into the system. But on the right-hand side, some of our assumptions, we were fairly conservative and said, let's say we get 85% moved from under-controlled, moving them from treated uncontrolled to treated controlled. Reduce office visits to about half and cut the number of meds. What does this ultimately give us? And it's staggering. I mean, we're talking about an aggregated savings for our treated uncontrolled of about $40 billion. So this is, again, on a per patient basis, is about $1,300 in per patient savings. So it's, I'm not going to harp on this too much, but it is, it's quite impressive, even if you can make, modest changes to the way this disease is treated. And we're not really introducing that a really small amount of cost is introduced through the genetic testing. So it's I would say not a huge lift on the front end and a big payoff on the back. And I am going to stop there. It looks like we're kind of bumping up against the Q&A session here. I wanted to make some acknowledgements. This is not everyone on the Geneticure team, but really important fact that a lot of us again come out of academia. Most of the folks, actually excluding myself, come out of Mayo Clinic up in Rochester. Eric Snyder, also a co-founder, much friendlier than his headshot may imply. But him and I met down here, and we're team teaching a class. And he did a lot of his training up in Mayo Clinic. He has a lot of physiology training, and also did his postdoc fellowship in nephrology and hypertension. Dr. Tim Curry runs the health education program for the Center for Individualized Medicine. And he serves as our medical director. Tom Olson, he's also still at Mayo, where he does see patients in the Division of Preventative Cardiology, again up in Rochester, also one of our co-founders. And then David Herbert, also out of Mayo. He ran Medical Mayo Labs for forever. He's since retired and joined our team. And then Scott is our kind of fearless leader here. He handles all things business. He comes out of the Target Corporation, also a co-founder with Eric, Tom, and I. And then Monica has been invaluable in getting us kind of up and running, handling a lot of our compliance issues and really doing a lot of the legwork. Carl has been fantastic in bringing us our operations together, making sure everything's moving properly. And Robin has done a lot of our IT work, also invaluable. So this is it's quite the team, and really lucky to be able to work with these folks, and I have to thank them a lot. Most of what I presented today is the product of their efforts, not mine. And with that, I want to say thank you for listening, and also again, big thank you to GenomeWeb and Agena. And I wanted to again put my email address up here and also our website. If there's any questions, feel free to reach out. I'm happy to again get some of our publications out if they're not available to you free. And then again, you can kind of go on our website and see if there's anything additional information you need. And I'll stop there. Thanks so much.
Great. Thank you, Dr. Sprissler. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to take a moment after the webinar has ended to give us feedback by taking our exit survey. All right, we'll now start the Q&A. Ryan, can you tell us what genes the 14 SNPs are in?
Sure. There's not really a trade secret there. We have here. Let me go back to the slide where we have those. So we're looking at CYP2D6. ADRB1 is the adrenergic beta receptor. So we have we have, let's see here. We have the adrenergic beta receptor 1, adrenergic beta receptor 2, cytochrome P450 is our CYP2D6. Excuse me. Lysine deficient protein kinase is the WNK gene. Sodium chloride symporter SLC12A3. We look at the alpha subunit of the epithelial sodium channel. That's the SCNN1A. We look at alpha adducin, ADD1. And then we look at the renin gene, angiotensin converting enzyme ACE gene. And so that's kind of the core set, and the 17 genotypes reside within those genes.
Great. How did you control for co-medication that could impact response to beta blockers because of inhibition or induction?
So I am actually, again, the molecular person. If I have some, I'm frankly afraid to answer that question. A lot of that was handled in the clinical trial protocols that were written by the other folks on our team, the clinical folks. So if you want to send that question via email, I'm happy to pass that on and get an answer there. But I'm sorry, I just don't. That's a little out of my depth.
So we have several questions about FDA approval for the test. Can you tell us, given how the FDA is now regulating the PGX space, whether or not the test is approved by the FDA, and if not, what plans you have to seek approval?
I'd say I'm glad you asked, but no, it's a yeah. We fully recognize this. We've been paying very close attention to this. Frankly, we agree with the FDA on many points, and we understand that the regulation needs to be there, and we want to make sure again that the whole goal was to generate clinical evidence and kind of. And we are looking to seek FDA approval, and we have been in contact with them. We want, we're for our large prospective study, we're in contact with them. We want them to comment on our trial protocol. We want to know what endpoints they want to see. We want to make sure that we're at least measuring those, collecting the data they want for those. So in short, yes, we're looking for FDA approval, and we're in the process of doing that through these clinical trials, and kind of building that evidence. In general, just as a personal comment, again here at the university we have a CAP-CLIA lab, and not all of our tests are varying levels of complexity, but I understand the FDA's need, and I understand kind of the frustration with it, and I think it's interesting to see this play out more recently with kind of the patient advocacy groups, but as a company, I think our main goal is to generate evidence, provide that evidence, and let that kind of speak for itself. And we kind of firmly believe in our discussions with the FDA have reason to believe that this is going to be the right path for approval.
Has the FDA given you any feedback on your choices of SNPs and the drugs that you make recommendations for?
I don't really want to get into the specific discussions we had with them and kind of their response to our questions and kind of the direct feedback we got. I will say in general, I didn't see any problems that they had with our variant selection. And again, all of the we're not advocating the use of off-label drugs. We're not, anything like this. We are strictly using the recommended therapies, again, just shuffling the order. So I don't want to speak to kind of those private conversations with them, but yeah, you can imagine if we're not going outside their treatment drug classes, it's likely not a huge hurdle.
Do you plan to offer updates for your reports after, say, AI learning or new scientific findings?
Very, very good question. I think you almost have to in some sense, and I guess it would depend on impact. So we by no means think that this is the end-all be-all for this particular panel. Other things are, I kind of look at this as internally this is like version 5, but I kind of look at this as version 1 with regards to content of the panel. As we add to it, we're going to have to revalidate. There's just such a huge process in kind of going back, and especially with regards to the clinical annotation and this trial. So yes, there will be future versions, and again, we're learning, and some of the other disease states we're working on. We have a number of other tests in the pipeline here. We're kind of learning how best to bring those forward and add or remove content, and then how best to validate it and kind of lean on generate the clinical evidence to support it. But yes, absolutely. If there is a major upgrade, breakthrough or significant change to the algorithm that has a dramatic impact to the patients, we would definitely put that out to them in an attempt to contact.
Can individual people order the test? And if so, do you have any telemedicine available to help them with reviewing the results?
At this point, we do not. Again, due to kind of the regulatory climate, there's a lot of these kind of, I would call them pseudo direct-to-consumer avenues where there's kind of a physician-mediated ordering where the patient orders a test and then it's that order is sent to a physician who can order it on their behalf. We think those are viable means of this type of testing. However, right now, due again to the kind of regulatory climate, we're a little cautious on kind of that pseudo direct-to-consumer channel. And so, in general terms, I would say no. This needs to be something your physician orders.
And can you tell us how you're handling the return of results to patients given recent FDA actions regarding pgx testing?
Sure. So again, we're very, very, very early in the process, but the report is meant for the physician. So the return of results goes through a physician.
And are you working with any reference labs, or are you planning to work with any reference labs in the future to offer the test?
So long term, that's definitely, right now, CAP-CLIA Environments is an LDT, high complexity LDT, so it's run out of one central testing facility. The answer is right now we are not, but yes, absolutely. I think, it just makes sense to have, an eventual kind of a decentralized testing system where you have a number of different reference labs. My preference would be to have this assay run in all molecular pathology labs across the country in various hospitals or anywhere that's being prescribing hypertension medication to patients.
Great, thank you. And we have reached the hour, so we're going to wrap up there. We'd like to thank Ryan Sprissler and our sponsor, agena-bioscience. If we didn't have time to get to your question, we will try to follow up with our experts. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. And if you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.